Welcome to the Eye on the Future Forum Spring Virtual Summit 2021. We will get started very shortly with our CEO, Daniel de Boer. But firstly, I would like to bring your attention to the live captions available that will be running alongside this event. You can access these by turning on the live transcript at the bottom of your screen. I will now hand over to our CEO, Daniel de Boer.
Thank you, and good morning and good afternoon, everyone, and welcome to this Eye on the Future Spring Virtual Summit 2021. We're happy that you could join us today for an exciting program that's featuring our clinical development work for USH2A Usher syndrome and retinitis pigmentosa. During the presentation today, there may be forward-looking statements, which are on this slide. The Eye on the Future Forum was created by ProQR to serve the community of patients, caregivers, advocacy groups, and healthcare professionals. And our goal is to bring each of these vital groups together into an ecosystem of people that are invested in treating inherited retinal diseases, understanding the clinical, emotional, and social impact of these diseases, and working together to develop treatments. The Spring Summit represents an opportunity for us to share important new data for our program for people with USH2A mediated Usher syndrome and retinitis pigmentosa, to continue to create meaningful dialogue, to further our understanding, and also to support connections. To this end, we've brought together an exciting group of speakers to highlight the work being done in this space. Mani Iyer was born and raised in Bombay, India, but lived in the United States since 1985. Mani is deafblind due to Usher syndrome type 2, and he was an engineer for over 30 years that has worked in several companies. After retiring due to blindness, he volunteered for the Usher Syndrome Coalition and developed and launched the USH Trust. Krista Vasi serves as the executive director of the Usher Syndrome Coalition and has been with the coalition since its inception in 2008. Since that time, she has led three main initiatives: forging relationships with researchers dedicated to Usher syndrome, identifying and strengthening the Usher syndrome community worldwide, and building an international registry exclusively for individuals with Usher syndrome. And Dr. Paul Yang is one of the top experts in the inherited retinal disease field and is currently an assistant professor in ophthalmic genetics and immunology at the Casey Eye Institute of Oregon. You can find more information and background on the panelists at the EFF website. So there are more than 2 million people in the world that live with an inherited retinal disease, often affecting children early in their life. These are diseases that lead to blindness in the retina, so in the back of the eye. Very few of these patients, less than a few thousand, have a treatment option available to them. All the other patients have no treatment options and are destined to become visually impaired. We believe that our technology can lead to medicines for approximately 25% of the mutations that cause inherited retinal diseases, and possibly more. We are committed to building out our platform to develop medicines for these conditions. To that end, we have built a pipeline of medicines that each target individual mutations that lead to these diseases, together forming a very sizable group of patients that we may be able to help with medicines that come from this pipeline. As I mentioned on a previous slide, our technology can potentially lead to medicines for a wide number of different mutations that cause inherited retinal diseases. These RNA therapies target specific mutations and don't need a viral vector or don't need to change the DNA to have their effect. The frequency of administration of all of our therapies is expected to be twice a year. Our pipeline currently consists of a platform for RNA therapies that has been steadily maturing over the last few years. The sepofarsen program for LCA10 has shown very positive clinical data, as well as QR-421a for Usher syndrome and retinitis pigmentosa. We have one more candidate in first-in-patient trial, which is the QR-1123 program for autosomal dominant retinitis pigmentosa in the Aurora trial. And a fourth program for Fuchs endothelial corneal dystrophy will start a clinical trial soon. And beyond that, we have dozens of earlier stage programs that could enter clinical trials in the years to come. Due to the generalizability of the behavior of these molecules, we are developing these drugs as a platform. Molecules that are so similar that the learnings on each molecule inform the next molecules in the pipeline. That makes us believe that we have a platform with a very scalable opportunity across many of the inherited retinal diseases. So finally, I want to quickly mention the sessions for today. We have an Usher program update by Dr. Aniz Girach, and then a panel discussion on the Usher clinical trials and the IRD community perspective with Dr. Yang, Krista Vasi, Mani Iyer, and Dr. Aniz Girach as panelists, which will be then moderated by Jennifer Plume. So now I'll give the word to Aniz Girach, our Chief Medical Officer at ProQR.
Thank you, Daniel. It's a pleasure for me to be able to present details about our Usher programs in the ProQR company. I shall be covering a little bit of information on the QR-421a drug, as well as a little bit of background information on Usher syndrome and non-syndromic retinitis pigmentosa. And then finally, and most importantly, I'll be covering information on the Stellar trial and some updates on the data and the recent interim analysis that we have performed. And so if you move to the next slide, please. It's known that the most common cause of Usher syndrome and non-syndromic retinitis pigmentosa are mutations on exon 13 of the USH2A gene. Now, this leads to a defective usherin protein. And this protein is needed at the back of the eye for the photoreceptors to be able to be functioning in a healthy way. Now, without that usherin protein, the photoreceptors, it leads to photoreceptor death. And that ultimately leads to blindness, which is characteristic of these diseases. QR-421a is an antisense oligonucleotide and is a first-in-class RNA therapy, and it's designed to produce a functional copy of that usherin protein. We know that there are about 16,000 patients in the Western Hemisphere that suffer from both Usher syndrome and non-syndromic retinitis pigmentosa. And that's likely to be probably an underestimate as well. QR-421 aims to prevent these patients from going blind. We were very glad to have some co-funding from the Foundation Fighting Blindness towards the conduct of this study. And if we move to the next slide, please. Usher syndrome and non-syndromic retinitis pigmentosa are really slowly progressing diseases that ultimately lead to blindness. They start presenting themselves in the early teenage years with night blindness. And this progresses slowly over a period of decades, leading to progressive visual field loss. And then ultimately, that visual field loss leads to a pinpoint of visual field left. And that in itself will also diminish such that even that central vision diminishes and patients go blind. And so we've thought about this disease in two categories. We've thought about the early to moderate stage of the disease, where there's visual field loss occurring, and then the later stage of the disease, the advanced disease stage, where even the central vision is affected and patients ultimately go blind. We know that the progression rates can vary from patient to patient, and therefore we think that the best control to use in any trial setting is probably on the untreated eye. And if we move to the next slide, please. The progression of Usher syndrome and non-syndromic retinitis pigmentosa is depicted in a visual illustration here. We have got two graphs, one in a pink color, which is depicting the visual field loss that occurs over a period of many years, slowly, and then ultimately leading to a very narrow visual field. And then at that stage, the degeneration that's going on at the back of the eye also affects the central cone photoreceptors, and therefore leads to a decrease in vision, central vision, known as visual acuity. Now, why is that important? It's important because when you think about visual fields or progressive decrease in your ability to see your peripheral vision, then that's measured in a clinical trial using specific endpoints or outcome measures called perimetry. And we'll talk more about that at a later stage. When the central vision is affected, that's usually addressed with an outcome measure called best corrected visual acuity. And again, we'll talk about that in a little bit later slide. And therefore, we're thinking about these disease in this early to moderate stage and the advanced stage. And I think that's a relatively easy way to think about the clinical trial data that I'll share with you later on. And if we move to the next slide, please. So the STAR trial is a first-in-human phase 1/2 trial, which is a randomized, sham-masked, single ascending dose global study. We covered 3 different dosing cohorts, 50 micrograms, 100 micrograms, and 200 micrograms. And we performed an interim analysis about a year ago, which showed that the drug was well tolerated at that stage and showed evidence of target engagement. And now what we have seen is, as a result of this final interim analysis, the outcome after 3 months of the last patients having been dosed with the 200 microgram dose. And we will see that actually this interim analysis had a different purpose compared to the first interim analysis. The goal of this particular interim analysis was to identify what to do next, what the trial would be like in subsequent time. And in particular, we were thinking of looking out for a possible registration endpoint. We wanted to establish the ideal dose and dosing frequency of the QR-421a, and of course, look for the ideal target population to be able to perform the next set of trials in. And so if we move to the next slide, we'll start to dive into the details of the data itself. So altogether, 20 patients have been enrolled into this study. Out of those 20 patients, 14 have had treatment with QR-421a, and 6 patients had sham treatment. And sham treatment was there purely for masking purposes. Out of the 14 patients treated, 8 patients had the early to moderate phase of the disease, 6 patients had the very advanced stage of the disease. And we will present data which includes at least patients who have had a 12-week follow-up. And you'll see that we will also include data up to 1 year time point or 48 weeks time point, because that's the time point when we have still a reasonable number of patients. Therefore, we can actually make some meaningful conclusions out of the data. We do have data that goes out even beyond that, right out to 2 years. But unfortunately, the number of patients that have got further follow-up beyond 1 year diminishes quite rapidly. The table below summarizes that QR-421a treated groups are reasonably well balanced, as opposed to the sham group patients where there is a little bit more imbalance going on, and therefore again highlights the importance of the fact that it may be better to use the fellow untreated eye of a patient as a comparator. And so if you move to the next slide, please. And so let's discuss the data, the safety first. So it's shown so far that QR-421 is observed to be well tolerated. And this is after 3,700 subject follow-up days across 2 years of follow-up. Importantly, we have not seen any serious adverse events, and there's been no evidence of any inflammation. We know that cataracts do occur in the natural history of this disease, and in fact, up to 50% of the patients may have cataracts. We did have 1 patient who had worsening of pre-existing cataracts in both the treated eye and the untreated eyes, and needed cataract extractions in both eyes. This was not deemed to be treatment-related by the investigator. Another feature, which is part of the natural history of the disease and can occur up to about half of the patients, is cystoid macular edema, or fluid at the back of the eye. There were no new occurring cases of CME during the study, and 1 patient did have CME at baseline, which progressed during the study. This was classified as mild and is being managed with standard of care. And so if we move to the next slide. So we've talked about the safety of QR-421a. What about the summary of the rest of the efficacy of the phase 1/2 results in this interim analysis? Well, I'm glad to report that clinical proof of concept has been established, consistent with the baseline disease. And then remember, this is after a single dose of QR-421a. Importantly, in the advanced disease population, so those patients who have a worsening of central vision already at baseline, we had all of the patients in a responder analysis who responded to the best corrected visual acuity endpoint, as opposed to none in the sham group. When we look at the entirety of the group in the endpoint, which is the best corrected visual acuity, and if we look at the mean values, there was a benefit in the QR-421a treated eyes by about 6.5 letters compared to the fellow untreated eye. And if we look at the advanced disease population, which is the correct disease population to look at because they are more likely to have central visual acuity loss, then the difference between the two arms was up to 9.5 letters. And the test-retest variability in best corrected visual acuity in this particular trial was 4 letters. And therefore, having a 9.5 letter difference between the treated eye and the fellow untreated eye is really very clinically significant. Now, if we then turn our thoughts to the early to moderate population, these are patients who had relatively good central vision, but nonetheless had relatively advanced disease stage because their peripheral vision was already collapsing in. In those patients, we actually found that there's an improvement in static perimetry. Now, static perimetry is a way in which we can actually monitor and analyze the ability for a patient to be able to see in the peripheral vision. And this is done using various instruments which actually shine bright lights into different parts of your peripheral vision and be able to then pick up the sensitivity at which a patient can actually pick up whether they can see the light there or not in the peripheral vision. And that's known as static perimetry. And we have actually now, for the first time, I think, shown that actually a drug can have a beneficial effect on static perimetry as an endpoint. And indeed, when we looked at one of the FDA potential registrable endpoints, then actually we found more patients had a benefit in that in the treated eyes compared to the untreated eyes. And that again fills us with a lot of confidence about that particular endpoint to be used as a primary endpoint in future clinical trials. So we had a benefit on central visual acuity in the advanced population. We had a benefit in the ability to improve the peripheral vision as measured by retinal sensitivity in the early to moderate population. But we also had concurrent improvements in other endpoints which moved in the right direction as well. Now, there is an endpoint which we tend to use, which uses optical coherence tomography, and that images the back of the eye, the retina. It gives us a cross-sectional view of the retina itself. One of the layers within that retinal image is called the EZ layer, or the ellipsoid zone layer. And the importance of that is that that layer signifies viability of photoreceptors. And therefore, it's very important to be able to preserve that for as long as we can, because that's usually a sign of a person being able to see versus a person not being able to see. And indeed, the FDA recognizes that as a registrable endpoint as well. The importance of OCT imaging is that it's an objective measurement, it's an objective evaluation of what's going on. And we found that actually in the QR-421a treated eyes, there was a stabilization in this EZ line, EZ area. And that's very important compared to a gradual deterioration in the fellow untreated eyes, which is what you would expect to see as part of the natural history of the disease. Furthermore, we had a similar stabilization shown in the endpoint called microperimetry, which is a form of perimetry which measures retinal sensitivity in the central 20 degrees of vision. And that again is another way of testing how well a patient can see in the relatively central zone of vision. So again, these are consistent endpoints moving in the right direction and also supporting the key endpoints of best corrected visual acuity and static perimetry. And these fill us with a lot of confidence that there is indeed proof of concept established and target engagement. We have also established that actually all of the 3 doses had relatively good efficacy. And that's indeed in keeping with oligonucleotides as a general rule. Oligonucleotides have a sharp dose curve, dose response curve, and they tend to stabilize with a relatively prolonged treatment effect as you increase the dose. One further element that we've established is the dosing interval also. We have established that approximately 6-monthly dosing interval is going to be the ideal timing for subsequent multiple injections of the drug. Remember, this particular trial only had 1 injection given at the start of the trial. So we have now all of the information required from the Stellar trial to begin to design our next stage of studies, which are the phase 2-3 studies. These will be called the Sirius study and the Celeste study. And if we move to the next slide, please, I'll describe these in a little bit more detail. But of course, before I do that, a very big thank you to all of the participants and caregivers and supporters who have helped with making the Stellar trial a success. Of course, this would not have been possible without the investigators and all of the site staff that were there. And of course, we have the wider QR-421a team, which also helped with this. So a big thank you to all of you for enabling us to really make this monumental step forward in research in Usher syndrome and non-syndromic retinitis pigmentosa. So let's just quickly describe the future trials that we're aspiring to do by the end of this year. These will be pivotal registrational phase 2/3 studies. Two of them. The first one is going to be called the Sirius study. This will be in the advanced patient population. The preliminary design has been discussed with the regulators, but of course, we need to also submit the protocols for formal validation and agreement by the regulators. But in principle, at least, we have agreed that this trial will be a 3-arm design trial, a target registration dose of QR-421a, supplemented by a second dose, because the regulators like to have a second dose purely from a masking point of view, and then also a sham procedure arm. And this 3-arm design is very typical of inherited retinal disease designs in the pivotal trial status. The trial itself will be a 24 months duration with an endpoint at month 18 and with the dosing occurring every 6 monthly. We'll approximately enroll 100 patients and we will have best corrected visual acuity as the primary endpoint. Of course, we will look at many other endpoints as well as secondary endpoints. And the anticipated trial start is before the year end. And if we then move on to the Celeste study, the Celeste study is very similar to the Sirius study, except that we're looking at the early to moderate patients here in the Celeste study. The trial design, like I said, involves a 24-month duration of follow-up with an 18-month primary endpoint. And like the Sirius study, there is a potential for a 12-month interim analysis. Approximately 100 patients will also be enrolled in this particular study, with the primary endpoint here being static perimetry, as opposed to best corrected visual acuity in Sirius. And of course, we'll be looking at other endpoints as well. And so if you go to the next slide, please. I think I will conclude here at this stage and hand over to Jennifer Pluin at this stage.
Thank you, Aniz. So this is Jennifer speaking, and I'd like to extend a warm welcome again to our distinguished panel, Dr. Aniz Girach, Dr. Paul Young, and Dr. Mani Iyer and Krista Bazzi. So for the next 60 minutes or so, we'll have an interactive panel discussion on Usher syndrome clinical trials and the IRD community, alongside answering any questions that the audience may have. Now, if you'd like to submit a question, please do so at either the Q&A and chat functions on Zoom, or also email eff@proqr.com. Also, I'd like to let you know that we have captions enabled, and you can turn these on at the bottom of your Zoom. And for the panelists, please feel free to pose questions to each other as well. We'd like to make this as interactive as possible. So let me just start off with some general thoughts on the Stellar results. The question is, what does the news in late March and shared today mean for the Usher community? So Mani, maybe I can start with you. What do these results shared today mean to you?
Hi, thank you, Jennifer, and thank you to the panel. As far as I'm concerned, I don't know whether there is any chance of an improvement in my case, because I just have light perception. But for all those people who still have central vision, especially the younger population, I am really excited. I think this is a fantastic finding that Dr. Aniz talked about. I say fantastic, and I'm excited because if the central vision can be arrested, then they don't have to go through the process of adaptation. The problem with Usher syndrome is because it degenerates at different rate, you have to constantly adapt here. So I'm thinking like if the central vision is arrested, then they just have to adapt one time and that's it. They can work around their limitations and continue with their life. Unfortunately for me, I felt that when I lost it completely, I lost my eyesight completely, because that meant I had to stop adapting anymore and get used to whatever I have and work around it. It was a big relief for me. But imagine if they could do that at an earlier stage when they still have vision. Wouldn't that be fantastic?
Thank you, Mani. Thank you for sharing that. That was, very heartfelt, and I really appreciate that. Krista, let's maybe pass it on to you. What are your thoughts on what do these results mean?
Thank you, Jennifer. Well, really, until recently, very few companies have been aware of Usher syndrome, let alone actively working on treatments for the Usher community. And to then reach a milestone like that of the Stellar trial is very exciting and energizing. And while the community, Mani, spoke to this, understands that this trial is for people with a very specific mutation, it still gives hope that this will pave the path to treatments for their own type of Usher syndrome as well. And that's the whole reason the coalition exists. This is why we've been building the global community and the Usher Trust Registry to be ready for this moment and ready to advance clinical trials by bridging the gap between researchers and the community. So we know this news is sending waves of hope and enthusiasm throughout the community.
Thank you. That was very nice. Dr. Yang, as a treating physician, what do these results mean to you?
Well, I agree with what Mani and Krista said. The data from the early phase clinical trial is truly encouraging. And I think, it does give hope to all the patients with Usher syndrome that, one day there may be an approved treatment for at least one type of Usher syndrome. And, if the treatment truly slows down or stabilizes retinal degeneration, as Mani had mentioned, it would really mean adding additional years of better vision and quality of life for patients.
That's great, thank you. So the next question, in terms of peripheral vision, did people see an improvement? I think you did show that, Aniz, but how do we measure this in clinical trials? Maybe you can just mention that again.
Yes, and I'll also ask Paul to comment as well. In clinical trials, we want to be able to look at peripheral vision in a number of different ways, but those are summarized in what are known as perimetry measures. And one particular way of looking at perimetry is something called static perimetry, where we shine bright light of different level of intensities of light at different parts of the patient's peripheral vision and see if the patient can pick up that light at different light intensities. And that gives us an idea of whether they can see or not in those spots, or actually whether there's an improvement or the same or decrease in the vision and the quality of the vision that's there. And that's a useful way of picking up what happens to the peripheral vision. And especially as the degeneration is caving in, it gives us an idea of whether there are seeing areas or unseeing areas and what's happening to those particular areas of sensitivity in the retina.
Dr. Yang, anything to add to that?
I think the only thing I'll add, Aniz explained it very well, is I'll also add that there is also the criteria of what it means to have a meaningful response. What's a meaningful change in visual field. So the generally accepted industry criteria for a meaningful increase or decrease is a change of at least 7 decibels, and decibels is a unit of retinal sensitivity that we apply or use for visual fields. And we need to see at least a change of 7 decibels among a minimum of 5 loci or separate data points.
Great, thank you. So this is Jennifer speaking again. Along those lines, stabilization of visual acuity is something we saw in the research. And what does this mean to the Usher community? Maybe, Mani, if I can, I think you mentioned it before, but is stabilization of vision an acceptable outcome for individuals affected with Usher syndrome? Or is it just gain in vision? Is stabilization enough?
I think it is the most important one. I mean, compared to having peripheral vision or night vision, and I mean, stabilization of central vision means a lot when it comes to adapting. Like I had to, I love books, and I used to be able to read paperbacks, and it was a practice of mine to read every night for 1 hour before I went to bed. And over time, I slowly realized that I was not able to read it at a distance I was used to. I had to bring the book closer and closer, and then I switched to large print books, and that also proved to be very difficult over time. And then I had to switch to audio. So things like that, every time you have to do the adaptation and work around it. And I think central vision is really, really important to preserve. I have one question for Dr. Aniz. He didn't mention much about the night vision part in his presentation. Did the QR trial do anything to night vision? Did it make any difference to night vision in the patient?
Mani, that's a very important question, because of course,
it will certainly improve the quality of life, just preserving the central vision.
Right, and just to answer your question there, Mani, we took patients who are relatively advanced, as it were, in the disease process, and therefore we didn't have any patients who are at that very early stages of the disease of having night vision problems to start with. But indeed, that's a very important area, because of course a lot of patients do suffer as night vision problems as the first symptom along the line of Usher syndrome.
Thank you. Kristen, you have any thoughts on the stabilization of vision question?
Yes, I'd really echo a lot of what Mani said. It's, I think for so many, that is really significant. Mani talked about the adjustment. It's a constant grieving process and a constant need to adjust and adapt because of the progressive loss of vision. I was talking with a mom about this recently, and she said if she could have that for her daughter, stabilize the vision she has left, that alone would be a huge win. So I do think it's a great start. That would make an incredible impact on so many.
Great. Actually, there's a question that came in that I think since we're on this topic, I'll ask now. And it's for USH2A individuals. How does someone with a genetic test know if they have this exon 13 mutation? Maybe I'll pass that to you, Dr. Yang.
Yeah, certainly the first thing is having had genetic testing done in order to even know that you have a mutation in USH2A. So as part of the genetic testing, the mutation is defined by its location on the gene. And one section of that gene is called exon 13. So by definition, if you've had genetic testing, then we know exactly where your mutation is and whether or not it's within the exon 13 section of the gene.
Great. Thank you. So again, Jennifer speaking, maybe we'll move on to some outcome measures. And what is the significance of one line gain versus a one letter gain? Dr. Yang, maybe if you can address that, one line gain versus one letter gain.
Certainly. So one line gain is equal to a gain of 5 letters. So one line, right, is a greater change than one letter. And where this is important is when, especially in a clinical trial, there needs to be a demonstration of that any perceived change is real and beyond the threshold of variability, which Aniz had mentioned that it's approximately 4 to 5 letters, in these patients. So that's really important to show that you have a signal that goes beyond just baseline variability.
Great. And Aniz, any thoughts from a regulatory acceptance point of view?
Yeah, it's really building on what Paul has just mentioned here. I think that there's one threshold, which is thinking about the retest variability of a particular assay or test that you're doing. In our trial, for example, it was 4 letters on the best corrected visual acuity chart. And so anything beyond that is we can usually get a feel that that's actually a meaningful change. Now, as far as the regulators are concerned, when we think about other disease areas, such as wet age-related macular degeneration and diabetic disease, there are some established thresholds of improvements or loss of a certain number of lines on the ETDRS chart. And typically, if you're working with the FDA, US FDA, the FDA like to see a 3-line gain or a 15-letter gain on the ETDRS chart. European authorities are a little bit more easier going. They tend to accept a 2-line gain or 10-letter improvement as clinically significant in their clinical trials. But those are importantly in other disease areas. In the inherited retinal disease space, we've really not fully established what clinical significance is. And of course, if you think about what Manny said and Krista mentioned, at the patient level, even a preservation of keeping the vision exactly the same is a meaningful outcome. And therefore, of course, vision gain is actually a real bonus at this stage. And so really preservation of vision and stabilization of vision is really what we're aiming for. And in our data set, we saw that in the QR-421a treated eyes, we had a preservation of vision there, whereas the untreated eyes fell away with their vision in keeping with the natural history of the disease.
Okay, great, thank you. Can you explain the difference between the different outcome measures used and why we use them? For example, best corrected visual acuity and static perimetry. So Aniz, this one's for you. Can you explain why we use these different measures?
Yeah, it's important to use different outcome measures for different stages of the disease. And it's because Usher syndrome and non-syndromic retinitis pigmentosa is a chronic disease. It's a slowly progressing disease over many decades that patients have at the beginning night vision problems and then visual field loss. At that time, their central vision is actually well preserved. So if you were to measure best corrected visual acuity in that patient who has just peripheral vision loss, then that best corrected visual acuity, which measures the central zone of vision only and utilizes the cone photoreceptors only, then that will be relatively well preserved and will be relatively uninformative for us in clinical trials. And therefore, we've got to look at specific outcome measures that address the specific changes that are going on in the peripheral vision. And in our case, it's using static perimetry, which is the gold standard for looking at visual field losses. Perimetry is then the standard that's used. As the disease progresses to the end state, when indeed the central vision is affected, then we use the gold standard way of looking at that particular outcome measure, and that's best corrected visual acuity.
Okay, great, thank you. Dr. Yang, any thoughts on what you assess on a regular basis for your patients? I'm assuming that within clinical trials are one thing, but then in your routine practice, do you do the same?
Actually, we do. In clinical practice, we use many of the same measures, and it's for the same reasons that was mentioned by Aniz. We use a combination of visual acuity, different kinds of static perimetry to follow the progression of the disease. And as Aniz mentioned, each type of outcome shows more change during different phases of the disease. And then we also use optical coherence tomography, OCT, to allow for an objective measure of the retinal structure of the photoreceptors in the macula. And we do that also to check for and monitor for any cystoid macular edema, which is something that patients with RP are at risk for.
Okay, great, thank you. Aniz, can you explain why we treat just one eye and have sham arm for trial as well? Isn't the comparison to the untreated eye enough?
Yeah, that's a very good question. So we treat the first eye, which is one eye only, because of course this we're still in experimental phase. For example, Stellar trial is a first-in-human study, so it's very experimental at this stage, and therefore we don't want to expose both of the eyes to treatments. So typically we would only treat one of the eyes, and typically also it's usually the worst of the two eyes, but not always. Now, as a result of the fact that regulatory agencies require us to mask studies to ensure that there is no, they try and limit the amount of bias that comes into the interpretation of the data. They want us to also include sham arms as well, independent arms where there is a sham procedure which is mimicking an injection, but actually there's no penetration of the glove. And that way we can eliminate any perceived bias that could be there. So those are usually the ways in which we perform clinical trials. In our case, because this is a phase 1, phase 2, first-in-human study, it's quite reasonable to be able to use the fellow eye as a control comparison, because of course we don't necessarily need to show regulatory level of acceptance, but this is the first stage of development to that particular process. We used sham patients in this particular trial for masking purposes to really enhance the integrity of the data that we have. And that's, I think, helpful to be able to understand the true value of this particular data. But indeed, as we move forward, as you saw, our phase 2/3 trial designs will include a sham control arm independently, and that's mandated by the regulatory bodies as a whole.
Yeah. Okay, thank you. Dr. Yang, a question for you. Is this a difficult concept to explain to your patients who may want to participate in a clinical trial?
I think it just takes time to kind of explain it. And I think when you think about it, it's logical that this is something that just has to be done. As Aniz said, there are pros and cons to each type of comparison. When you're comparing the treated eye to the untreated eye, it allows one to control for the patient's disease state, whether it's early to moderate or advanced disease. It's relatively symmetric between the two eyes. Whereas a sham group helps one to control for subconscious bias that any patient may have when they're enrolled in a treatment clinical trial. So really, you can't really have a good clinical trial with outcomes that FDA can use to decide whether a treatment should be approved if you don't have either an untreated eye or a sham group. So it's really necessary, and I try to explain that to patients in a way that being in the sham group, even though you're not being treated, is just as important as being in the treated group, because without the sham group, there would be no approval of the treatment.
Great. Thank you. Yeah, very important. Aniz, a question for you. In the early to moderate population of Stellar, why do we see an improvement in static perimetry, but just the stabilization on best corrected visual acuity? Why doesn't the visual acuity improve in this group of patients if they're less severe?
Yeah, that's a very important question. It really all boils down to the baseline disease stage that the patients are at. So for example, in the early to moderate patient population, even though we call it early to moderate, actually that's a misnomer, because for those patients, remember, this is a chronic degenerative disease over many decades. And so for those patients, the degeneration is actually very active at that area in the peripheral, mid-periphery of the patient's vision. And therefore, the cell loss and cell death that's occurring is actually very advanced at that particular area. So those patients have a very low threshold. They've lost a lot of function in many of those photoreceptor cells. And therefore, I think there's a potential for us to be able to improve that function in those cells. There are various theories like the transition zone theory and other things that are branded out there. But I think it's to do with the fact that this is all to do with the low baseline there. Now, as opposed to the visual acuity, in this particular trial, the baseline visual acuity was around 20/40 or 20/50, which means that patients had only lost 3 to 4 lines of vision at baseline. So there are many patients who had relatively good vision, and therefore there's a ceiling effect. And so it's unlikely for us to be able to see any big improvements in central visual acuity because of that ceiling effect. And therefore, it's probably much more appropriate to be able to see a stabilization of that relatively good central vision that's there over a long period of time. And that's what we saw in the Stellar trial interim analysis. And in comparison, the fellow eyes and also the sham eyes behaved in a similar way, where they actually waned away with the central vision in keeping with what the natural history of the disease is doing. And the QR-421a treated eyes had a stabilization of the central vision. So helps you to understand that it's really all down to the baseline disease stage at which they come in, and that determines whether they potentially could benefit from an improvement, or if there's a ceiling effect, that means that we can only keep a maintenance or stabilization of the vision.
That makes a lot of sense. Thank you. Dr. Yang, anything to add to that?
Yeah, I think maybe another way to think about that is, if we think about what is the contribution of the macula to the patient's vision, in its entirety. When we look at, say, electroretinography, we know that the macula contributes about 5% of the total signal. So we can kind of translate that to thinking about, what's happening in, so-called early to moderate disease versus advanced disease. So in early to moderate disease, when the patient is losing their peripheral vision, so basically all the areas that are outside of the macula, you can kind of consider, well, that's about 95% of the electrical signal. So as Aniz said, we don't want to downplay, the amount of loss that is occurring in early to moderate disease phase. That is still a lot of vision that's being lost. It's just that it happens to be outside of the macula. And during that period of time, the visual field at any moment has a lot of areas where it's defective. And so there is plenty of room for improvement. And I think that's why it's makes sense that you can measure improvement on static perimetry of the peripheral vision in early to moderate disease. However, the macula is typically not severely affected during that period of time. That 5%, that macular function, fortunately, that's not affected significantly until advanced stage of the disease. So what that means is there's actually a ceiling effect of visual acuity, of when you're monitoring visual acuity in the early to moderate disease phase. Vision can be quite good, and you can't really, get better than that during that period of time. And it's only until you get into the advanced disease phase where visual acuity begins to decline, where you then have room to see either perhaps an improvement or at least a stabilization of the treated eye, while the untreated eye declines as part of its natural history.
Great, thank you. That was very interesting and very enlightening. Dr. Yang, another question for you. How do research organizations and private clinics choose which outcome measures to use? Perhaps maybe your thoughts on what a standard clinic would use versus one that's a teaching institution or somebody who participates in research and clinical trials. Would they use something different or less than what's used in research institutions?
Sure. So I think probably the one thing that stands out as a difference in what is available in a so-called standard clinic versus a research center is the visual field perimeter. So these wide field visual fields that we've been talking about, they're performed on specialized machines that allow us to look at the peripheral vision all the way out to 180 degrees wide. So that machine is not available everywhere. And actually what's more common in ophthalmology is what's called a Humphrey perimeter, which allows an assessment of visual field anywhere from 10 to 30 degrees wide. So therein lies, one difference in that in patients with RP, in the early to moderate disease phase, you have a lot of changes going on in the mid to far periphery. But without, the wide field perimeters, you don't really get a good sense of what's going on during that period of time.
Great, thank you. Aniz, another question for you. With the positive announcement from Stellar, which was the phase 1 and 2 clinical development combined, what is your hope for the next stage? You mentioned the 2 trials, and believe you mentioned why, but if you could just tell us again why 2 trials and how many clinical trial participants will be needed.
So let's break that down. So what's the hope for the next stage? Well, what we want to do is ultimately, of course, confirm the findings that we've had so far. The findings we've had are in a preliminary study. It's an early stage study with low numbers of patients. And what we aim to do is in the phase 2/3 study, we aim to be able to then confirm those findings. So, we want to look at the safety of the drug. Is it still well tolerated in large numbers of patients? We want to look at the efficacy of the drug, and are we seeing the same level of efficacy as we've seen in the early patient populations that we studied. So that's really the purpose of the next phase of the trials. And then, of course, if the drug is safe and well tolerated, and there is efficacy seen according to the regulatory hurdles, then, of course, we have an opportunity to be able to submit the drug for potential approval to the regulatory agencies. So that's really the hope for the next stage of development. Why 2 trials? Well, it's really boiling down to what we have mentioned before, that there are different outcome measures that look at different stages of the disease. And so if you have that early to moderate stage of the disease, we'll be looking at perimetry as the main outcome measure. When we have a very advanced stage of the disease, then perimetry won't really help at that stage because it won't. Most of the vision field is actually lost by then, and therefore you have to look at a different outcome measure. And best corrected visual acuity is what we tend to use for those very advanced stage patients. And so these 2 different outcome measures, it's very difficult to be able to combine these 2 sets of patients into one combined trial unless you have a huge trial. But the disadvantage of that is that then you'll have part of the population having a ceiling effect, and another part of the population may not be able to have any effect on part of the endpoints. And so it's much easier for us to be able to then separate those 2 populations out and separate those 2 endpoints or outcome measures out into 2 separate trials. Each one of these trials could in themselves be enough for hopefully a registration trial in itself. And so depending on the validity of the data that's there. So I think it allows the company and for us to be able to have essentially 2 shots on goal at trying to provide some kind of therapeutic for patients with Usher syndrome and Boer syndrome.
Great, thank you. So Jennifer speaking again. Mani, next question would be for you. Is there something we can do at ProQR to make participation in clinical trials easier for individuals with an IRD? For example, some participants may have to travel far distances and find themselves in unfamiliar surroundings. How can we ease the burden for these patients? Do you have any ideas on that? I don't know, Mani, did you hear me? The question?
Oh, I know my internet connection is unstable. It keeps saying that. Can you repeat the question, please?
Yeah, sure. So I was asking if there's something we can do at ProQR to ease the participation in clinical trials for individuals with IRDs. For example, some participants may have to travel far distances to get to a clinical trial site, and they may be in unfamiliar surroundings. Is there any ideas on how we can ease the burden for these patients who have to travel far and wide for clinical trials?
I mean, there's a lot of anxiety around Usher syndrome patients. Anxiety is a natural cause of the disease. So I would recommend that make the patient as comfortable as possible and assure them that everything will be taken care of from point A to point B. That one thing. And then I don't know, just make them comfortable throughout the clinical trial. That's the most important thing.
Yeah.
But I'm sure a lot of people would be would agree to do it because there's a lot of desperation around it. Yeah.
Great. Thank you. You're right. There's a lot of anxiety produced with going into unfamiliar surroundings. Krista, anything that you want to add? Any ideas that you may have?
Yeah, so this is really such a diverse community in terms of accessibility needs. You'll really have to work with each person one-on-one to make sure that they have all the accommodations they need to participate. Might take a little bit more time, but it's going to be worth it. And the coalition can help ProQR know what you might expect. It could be a communication need, a support service provider to help with navigation, materials in large print or Braille. ProQR will need to recognize that this will be a very individualized process. And as Mani said, there's a lot that goes into venturing into something like that, but I think there's enthusiasm for people to participate. So
thank you. Yeah, I think it's good for us as a company to remind ourselves of the burden that study participants experience and focus on how we can ease this burden as much as possible. One of the patient steering committee members had a suggestion of maybe assigning a buddy so that you're traveling, the participants traveling to an unfamiliar city or new surroundings, assign them a buddy so that they can help them navigate the clinic and the hotel or whatever's needed. So I thought that was an interesting idea. But yeah,
I think so too. Yeah.
Yeah, it's very important to try to make it as seamless as possible.
Right. And I think that this is Krista speaking. That buddy system is a great idea. And I know that there's training available that person can get some guidance as to how to guide somebody who's visually impaired. And there's a certain way to do it and make sure that person is as comfortable as possible. So I think that'd be a great idea.
Yeah, great. Thank you. Krista, something else to ask you. If someone with Usher syndrome is searching for information regarding clinical trials, where do you suggest they search? What's the most reliable source of information?
Well, we do direct people to our website. We have a page of progress in research to treat Usher syndrome that covers both gene-specific research and trials like this one, and also progress in treatment development could benefit anyone, like stem cell therapy. So we really try to make sure that we have the most accurate and up-to-date information on our website. That's a place to start. Our registry as well, we encourage people to join the USH Trust, and that way they'll be informed of very specific trials that are relevant to their type of Usher syndrome, like the upcoming, Sirius and Celeste trials. Our annual conference is also a really great source of information and connection. It also often highlights some of the areas for future research. We truly identify gaps in research when we all connect directly. So that's another place.
Coming up in May, right?
Yes, the virtual conference in May, and hopefully next year we'll be in person again.
We all hope that, right? Mani, this is Jennifer speaking again. Where do you find the most reliable information?
So before I lost my eyesight, I used to go to the Foundation Fighting Blindness and keep up to date on all the latest research and all the exciting things that was happening around the retinal research. But then after I got involved with the Usher Syndrome Coalition, and I helped them develop the Usher registry, I have a direct line with Krista. So I can ask her what is going on, and the clinical trials, anything. Yeah, and she's really helpful in that.
She's a very valuable resource.
Very valuable resource. Yeah.
Yeah. Great. Thank you. So Dr. Yang, I think you mentioned a little bit about this earlier, but how important is genetic testing and the need for individuals with an inherited retinal condition to feel empowered about knowing both the clinical as well as the genetic diagnosis?
That's a great question. And so having a genetic confirmation of the clinical diagnosis not only allows for more confidence in the diagnosis, but I think it empowers the patient to have access to clinical trials that are specific to that gene and that mutation. It also allows for more detailed discussions on the prognosis, the visual prognosis based on prior studies of mutations in that gene.
Okay, great, thank you. Krista, a question for you regarding that. For someone who's reaching out to the Usher Syndrome Coalition for the first time and who doesn't have a genetic diagnosis, what advice do you give them?
Yes, we also really echo the importance of genetic testing and try to educate on if you don't have a genetic confirmation, you won't have access to clinical trials like this, and it really can give you insight into your own journey moving forward and help people make decisions for themselves. So we really try to make sure the why is understood. Why should I get genetic testing? And then the how, there's Foundation Fighting Blindness's open access genetic testing program now where people can get genetic testing for free. So we really encourage people to go take advantage of that. We're in touch with so many who have been living with Usher syndrome for so long and may have gotten genetic testing decades ago and think that they are there, but they don't understand. They never got the results because it was part of a study, or they don't fully understand their results. So we also encourage them to try to get that genetic testing confirmed with the most current technology, taking advantage of FFB's program, and again, making sure that information is in both the USH Trust and FFB's My Retina Tracker. So genetic testing is huge. Each week, I would say, our small team of 3, we hear from at least a couple new individuals in the community who are just opening their eyes to this diagnosis. And whether it's a parent or it's an adult who may have Usher syndrome, we truly handpick the resources that may benefit them. To be honest, it's almost overwhelming how much stuff we have to tell them because there is so much out there. There's so many resources. And then we also connect them with our Usher ambassadors, which is our network of volunteers who live with Usher syndrome and know the resources intimately that are available in their own regions. And the Usher ambassadors will also be those who help disseminate information about clinical trials like this, and genetic testing options. So we really try to send them every which way that can help with their journey.
Great, thank you. That was very helpful. And I think there was a question regarding genetics that I want to pose to the panel. Here it is. For USH2A individuals, how does someone with a genetic test know if they have this exon 13 mutation? Where do you ask that? Okay, so I'll go to the next question because I think we already answered that one. Will the next phase include the patient cohort that participated already or a brand new group? I'll pass that to Aniz to answer.
So the Celeste and Cirrus trials will include new sets of patients who will be recruited into that trial with specific characteristics that will fulfill the eligibility criteria. Now, however, the patients who are enrolled in the Stellar trial, what we will do is we will then roll these patients over into an extension trial so that these patients have the ability to still gain access to free drug and be monitored in terms of the safety and efficacy in a longer term setting. And indeed, in that extension trial, we aim to be able to give the choice to the physician and to the patients to be able to potentially treat the fellow second eye as well. And so that will really enhance the volume of data that we can gather in parallel to the pivotal trials ongoing.
Great, thank you. Is the RNA therapy only useful if you have some sort of remaining vision? Maybe I'll pass that to Aniz first and then get another viewpoint from Dr. Yang. So Aniz, for the RNA therapy?
Well, we don't know the real answer to that yet. We are exploring all of these avenues. So we're looking at patients with, the early to moderate disease and then also the advanced disease stage as well. Now, in terms of measuring outcome measures, of course, it's much easier when you have remaining vision that's there to be able to see what's going on with that. But we don't know the real answer to that. And I think that future clinical trials are going to inform us on, where exactly the RNA therapy can be of benefit.
Along those lines, maybe Dr. Yang, can you tell me if you think the community is aware of the difference between a gene therapy and an RNA therapy?
That's an excellent question. So there are differences. One thing, actually, there's multiple things that I think we should address. First of all, gene therapy is thought to be one and done. It's a permanent treatment, whereas RNA therapy has a half-life to it, and there's a need for retreatment, routine retreatment to occur. So that's one big difference. Another difference is that RNA therapy, as we've talked about, tends to be very specific to one type of mutation or a set of mutations within a region, like exon 13. Whereas gene therapy, and in its most common form, which is gene augmentation, where you deliver a full copy of the normal gene to the retina, it doesn't really matter what the mutation is, because you are delivering an additional normal copy of that to the eye. Now, I'm going to segue straight to the problem or the limitations of the vectors that are used in delivering a full copy of a gene to the eye. So the most commonly used vector for gene therapy is adeno-associated virus. But there is a limitation to how large of a gene that you can insert into the cassette for delivery to the eye. And the USH2A gene is simply too big to place in the AAV vector. And so there's a limitation there. And so RNA therapy gets around that. RNA therapy, has no limitation on how large of a gene you can treat, because again, these are small fragments of RNA that are designed to improve the way DNA is translated into protein. So it works very differently, and so it's not limited by the size of the gene. Let's see, Aniz, do you have anything to add to that?
No, I was just going to add how the RNA therapy is delivered. Typically, it's through an intravitreal injection, which is an in-office injection under local anesthesia, whereas most gene therapies, at least, tend to need a general anesthetic and are a little bit more complicated because they are trying to deliver the gene in a specific area under the macula. And then I guess for us, more importantly, since you administer the drug through the vitreous cavity, you can actually target the disease at a much earlier stage, like we've tried to do, which is to look at disease in the early to moderate phase of the development of the disease progression. Whereas many gene therapies are delivered through this subretinal delivery route, which means that they will inevitably have to wait until the end stage of the disease for them to have a beneficial effect. I think the technology is just not there at this stage to be able to target the entirety of the retina. Whereas I think that's an advantage of the RNA therapies, where potentially if the RNA therapy can work, it can actually arrest the progression of the disease to the advanced stages.
Great, thank you. Okay, next question from the audience. Will participants have their second eye treated in Stellar? I think you already mentioned that they're going to be moved over to the Helios study. And second part of that question, and will those who undergo sham procedure have access to the treatment? Oh, okay, maybe that's for the new trials, Sirius and Celeste. Will those who undergo sham procedure have access to treatment?
Yeah, so I'm not sure whether the reference is to do with the Stellar sham patients or whether it's to do with the sham patients that could be enrolled in the Sirius and Celeste studies. Either case, what happens is that typically, if you're in the Sirius and Celeste studies, after the primary endpoint evaluation is done, then most trials allow the provision of a crossover for the sham patients to be then actively treated with the drug dose. And that's what we anticipate on doing as well. It's exactly the same design that we've been employed with the cephalosporin and in the CA10 world and the phase 3 that's ongoing there at the moment.
Great, thank you. Yeah, I wasn't sure what the position of that was, but I think you answered both of those, so thank you very much. For the next phase, will there be more trial centers or will the current trial centers remain the same? Also, will geographical location of potential participants be a critical factor too? And that's for you, Aniz.
Yeah, very important question. So of course, we have some very important and prominent trial centers, like where Paul is in KCI in Portland, Oregon, which are hub centers where a lot of patients with inherited retinal diseases are followed up. And so we certainly want to encourage those centers to be part of our future trials. And we hope that they have the bandwidth to be able to participate in those 3 trials. But indeed, because we're looking at more numbers of patients, it's likely that we will expand the number of sites also. And therefore, we haven't exactly calculated how many sites we would need at this stage. There's active work in progress at the moment. Up to sites to see how many patients they have who would potentially fulfill the eligibility criteria. And so that's really work in progress at this stage. But it's likely that there'll be many more sites than the ones we used currently in Stellar.
Okay, great, thank you. Another question, Aniz, is again for you. Will it help in the, will this, will 421A help in the preservation of light perception? Like in my case, I rely on this light perception. So is there any, was there any data on preservation of light perception?
Well, in the current trial, these patients had relatively good vision at baseline. And so we did not explore patients who had light perception. Now, in theory, of course, if, and it's a big if the phase 3 trials are shown to do show that the drug is safe and is efficacious, then in principle, in those patients that have earlier stage disease, you can extrapolate for later stage disease as well. So I do think that there is a potential for patients at any stage of the disease to potentially benefit from treatments that could be safe and efficacious and are deemed to be safe and efficacious by regulatory bodies. But ultimately, it'll be up to the regulatory bodies to be able to decide whether they give a label or an indication that extends out for the entirety of the population or to subset the population into the ones studied specifically in those clinical trials.
Great, thank you. Okay, let's see. Oh, okay, there was a resource for European attendees that was shared. That's the ERN-I website, ERN-EYE website. It's a good source also of information. Question regarding the drug itself and the volume. What is the volume of the drug given by injection in the next phase and in Stellar? So maybe let's just overall, what is the volume of drug given by injection for 421A?
Yeah, so that's dependent a little bit on the dose that you use. But for example, in the Stellar trial, the typical injection volume was 50 microliters, which is in keeping with the volume that generally is injected into the vitreous cavity of most of these diseases and previous history. And that's quite a normal level of volume of injection.
Okay, question for Krista. Will there be research presented at the USH conference in May?
So yes, this year will be a little bit different from our virtual conference last year. We are doing more live sessions, and we will be centering around more perspective from the community and resources for the community. That being said, we are getting all of the content out that we shared last year and trying to get that out in more digestible ways so that people can get the research updates that are available to them in the past 9 months or so. And then we will have a live Ask the Researcher Q&A panel as well. So another opportunity to join on, ask these leading experts your questions about various types of therapies that are in development. So
excellent.
Please.
Yeah, looking forward to it. Okay. Here's a question. How important do you think it is for clinical trial participants to know if RNA treatments can change the genetic information? Can this then pass over to the next generation? I'm not sure who wants to tackle that one, but
maybe I can start, and I think Paul may be able to also find this. I think that may be related to some concerns about gene therapy and DNA editing, where there is a concern that could affect future problems with cancer or other things that could occur. And I think that as far as gene therapy and or RNA therapy is concerned, usually if you look at the gene therapy like AAV-based therapies and then also the RNA therapies that we have, they don't tend to affect the other cells in a bad way like that. And there's, I would say that the regulatory bodies have really looked at the data in its entirety and have opined on the fact that there's a very, very low chance of anything being passed on to future generations, and so it's negligible. So I think that's perhaps where the question is aiming for. Paul, anything else to add on that?
Yeah, I would agree with that. I think what we know so far in preclinical studies and in the clinical studies to date is that the treatments to the eye seem to mainly be isolated to the eye. Even preclinical studies that were done for gene therapy, one may find inactive elements of the AAV virus in the rest of the body, but really only just inactive elements and not actually live elements that can transduce cells. For RNA, it's probably even less likely, that it could affect future generations because these molecules have, as we talked about earlier, have a half-life to it, so they don't last for a very, very long time. As far as gene editing, such as CRISPR, that's still relatively new. So I think it will take time to collect, that sort of data. But again, because we are working with the eye, and the eye is an organ that is physically separated from the rest of the body, whatever treatments that we do in the eye tend to, stay isolated to the eye. And so that's kind of one fortunate aspect of, doing gene therapy for retinal diseases.
Great, thanks to both of you for that. Mani, this is Jennifer. Question for you. When you joined the Usher Coalition, why was it important to set up a registry?
Because we wanted to make sure all the researchers had access to Usher syndrome patients all over the world. That was our primary goal. And plus, it could be used to enable further communication between the Usher syndrome patients. That was our idea, but the primary goal was access to the researchers. At that time, I think there was no registry available. I don't know whether the foundation had something. I don't think, maybe Krista can correct me. I don't think the Foundation Fighting Blindness had one. So that was the reason. Yeah.
Okay, thank you very much. So Krista, here's the question for you. Do you work with organizations like ProQR to share your USH registry? And why should someone join? Well, the second question is, why should someone join the USH registry?
Yeah. So I'll add to Mani's answer as well. And, Foundation Fighting Blindness, my retina tracker, has been around for many, many, many years, but it is focused on all types of inherited retinal diseases. So the USH-TRUST registry is the first and only registry that is exclusively focused on individuals with Usher syndrome. And it's really the first time that the Usher community has been looked at as a unique entity. There's also deafblind registries. So there's other places that those with Usher might be a part of, but this brings everyone home together. And it also was created by Mani and the Usher Syndrome Coalition because it's so important that the community owns the registry data, and that it can be, to answer the question, it can be used in a secure and safe way, working with companies like ProQR and others who are in and entering clinical trials. That's its purpose, but it is always done in a secure and safe manner. And why join the registry? To be able to be part of something like this. And, it's always the registrant's choice. It's always just, we are informing the community what is available to them, and their information is never shared beyond that. It's on them to then make the informed decision to participate if they're comfortable. So.
Great, thank you, very helpful. So we only have a few minutes left, and so I think what I'll do is conclude by asking each of you if there are any final thoughts that you have from our discussion today that weren't already covered. So maybe I'll start with you, Dr. Yang. Any final thoughts that were not already covered in the session through the questions?
I think we did a good job here of answering all the questions. I think the data that has come out recently, I think it's very encouraging for all the stakeholders, the patients, the clinicians, the sponsors. I think it offers hope that treatment is possible, and that that platform, the antisense oligonucleotide RNA platform, can be a effective strategy for treating those large genes that just don't fit inside conventional AAV vectors. I do have one question that I thought of during the session, which is a question for Aniz. So if we fast forward into the future, Aniz, and hopefully we have an approved treatment for mutations in exon 13, how do you envision a practical strategy to develop treatments for other mutations outside of exon 13?
Yeah, great question. Of course, like you say, the RNA therapies are mutation specific. What we'd like to do, of course, as even Daniel mentioned in his earlier slides, is to be able to develop multiple targets and to be able to develop antisense oligonucleotide therapies against all of those targets. And so I think there's every reason and every possibility of being able to develop these kind of targets against each one of the different type of mutations that are out there, theoretically. And the technology exists now to be able to do that very effectively, very efficiently as well. And I'm hoping that, of course, as various diseases have therapies that are approved within that disease, that the regulators allow us an ability to more efficiently get further drugs approved with other mutations within the same diseases, which cause the same disease. And so I think that with some help from people like Krista and the Usher Coalition and other patient advocacy groups, and with the help of experts like yourself for influencing the regulators to be able to make life a little bit easier in performing these clinical trials and getting more therapeutics out there for patients, I believe there will be a stage where we can actually address many of these targets, many of these mutation types for these disease-causing mutations that are out there. So, only time will tell. I think that we're at a great stage at this stage to be able to get at least the main mutations addressed with this particular antisense we're looking at now.
Thank you, Aniz. Jennifer, if I may, I actually also have a question for Krista and Mani.
Yeah, sure.
So occasionally, I do have some patients that are worried about genetic information privacy, and they are worried enough that they forego genetic testing, due to these issues. And so what do you say to patients regarding this?
This is Krista speaking. We, first of all, acknowledge that fear and anxiety around that. And we certainly recognize that that's a valid thing to be concerned about. But when it comes to any information that the coalition is going to collect, we can assure everyone that it's completely secure, HIPAA compliant. We're not sharing their information beyond our own organization. And it's that's why we renamed the registry the USH Trust, because we know that the community as a whole has dealt with trust issues in the past and may feel they have been taken advantage of in the past. So we want to make sure that they know that this is a place they can come that is safe and secure. So the genetic testing, yeah, that's an understandable concern, but it's hopefully they will, with the assurances that we can provide, they'll move forward with getting genetic testing. And then more importantly, also having a knowledgeable genetic counselor who's familiar with Usher syndrome to help walk them through those results as well. Mani, did you want to add anything to that?
Yeah, when we took that into consideration when I developed the registry in the beginning, HIPAA compliance was very big for us. And when we made sure that if we had to give data to the researcher, we made sure that they just had the minimal information like the name and the contact address, and not depends off the genetic information and any other medical information. It was very, very important for us.
De-identified information is really all we would share at this point. So nothing that's identifying an individual.
Great. Well, thank you for that. So I think we're running out of time, so I think I'll close. But in closing, I first want to thank all of you as panelists for participating. It's been an honor to learn so much from you during this event. And to everyone attending, thank you for your time and commitment to the IRD community. And while there's so much uncertainty related to COVID-19, we look forward to seeing you at the next EFF, hopefully in person next time. But I'd also like to direct you to our websites. And I think there we are. They're displayed on the slides here. And here you can find information on the ProQR clinical research website, information about our trials, and also subscribe to our newsletters. We have 2 newsletters, Expert Eyes, which is our healthcare professionals newsletter, and iConnect, which is the newsletter for the IRD community. We publish these quarterly to keep you informed of our activities in the domain of inherited retinal diseases. And finally, just one last word. We try to make EFF as accessible to everyone as possible. However, there's always room for improvement. And we appreciate your feedback and suggestions on the format of the slides, the captioning, or any other subject related to the format of this event. And if you have any questions or comments, you please send them to the EFF@ProQR.com email address. So with that, I'd like to thank you all again and enjoy the rest of your day.
Thank you, everybody.
Thank you.
Thank you.